Hello everyone, and welcome to Lab Manager's Tech Trends webinar series. My name is Trevor Henderson, Technology Editor for Lab Manager, and I'll be moderating today's discussion, which will focus on clinical chemistry technology. We like our webinars to be very interactive, so we encourage you to send us your questions at any point during the presentation, and the panelists will address these during the question and answer session following the presentation. To ask a question, simply type your query into the Q&A box, which is located at the bottom left-hand side of your screen. We'll try to address as many questions as possible during our time together. However, if we happen to run out of time, I'll forward any unanswered questions to the panelists, and they can respond to you via email. Additional resources may be accessed by the resource window, and you may also move or resize any of the windows simply by grabbing them at the top or stretching them at the bottom right corner. At this time, you may need to move or minimize some of the open windows to see the live view. This webinar recording will be available early next week on Lab Manager's website, and at the end of this webinar, we'll share that link with you, as well as the contact information for our panelists. With that, let me introduce our first speaker, who joins us today from Thermo Scientific. Dr. Joe DeBussolo earned his Bachelor of Science degree in Clinical Chemistry at West Chester University of Pennsylvania in 1977. In 1982, he received a PhD degree in Biological Sciences from Drexel University in Philadelphia. Dr. DeBussolo then spent 20 years in the development, validation, and support of various analytical instruments and software products for the PerkinElmer Corporation. In 2002, Joe joined Cohesive Technologies and established a field applications laboratory at West Chester University of Pennsylvania. He has continued working there as a senior application scientist since Cohesive Technologies was purchased by Thermo Scientific in 2006. Joe is also an honorary faculty member of the university and participates in their pharmaceutical product development program. Joe is also a member of the American Chemical Society, the American Association for Clinical Chemistry, the American Society of Mass Spectrometry, and the Chromatography Forum of the Delaware Valley. Joe, thank you for joining us today.
Thank you very much, Trevor. We'll go to our first slide. And hello. This is Joe DeBussolo from Thermo Scientific, here to present ways to achieve the most efficient productivity when analyzing biological fluids using liquid chromatography with mass spectrometry for laboratory medicine. As you probably know, the interest in clinical LC-MS publications has increased exponentially over the last few years, which means that more and more clinical laboratories are considering or are already implementing LC-MS systems for various applications. This is because LC-MS technology offers great flexibility for lab-developed tests to measure clinically relevant compounds with confidence in small volumes of sample. The Merriam-Webster Dictionary defines the word efficiency as the ability to do something or produce something without wasting materials, time, or energy. Clinical laboratories must produce many analytical results of the highest quality as quickly as possible and as economically as possible. Our goal is to simplify routine workflows of preparing and analyzing many samples as efficiently and economically as possible within the confines of our laboratory resources. The design of compact multi-channel HPLC, tandem mass spectrometry, and software must ease our concerns for speed, efficiency, and laboratory space restrictions. Here we see Class 1 medical devices that were used to develop the example applications that will be shown. To the left, we have the Prelude MD to perform combined sample cleanup with HPLC in each of two channels that elute into one mass spectrometer. And we actually have an example mass spectrometer to the right of that, the Endura MD triple quadrupole mass spectrometer. Over to the right, we see the Prelude LX4 MD, which processes many samples across 4 HPLC channels, and they all elute into one mass spectrometer, such as the Endura. ClinQuan MD Software controls these instruments, processes the data, and generates reports of the results. The Prelude MD instrument is intended to separate drugs or compounds from a sample solution and introduce these separated drugs or compounds to a detector. It is for in vitro diagnostic use. The Prelude MD utilizes TurboFlow technology for online sample cleanup, which replaces offline solid phase extraction. This is coupled to HPLC or UHPLC, which separates compounds of interest and elutes them to the mass spectrometer. Doing this across two channels greatly increases sample throughput for one laboratory-developed test or permits you to simultaneously run two different tests which utilize the same mass spectrometer. So let's talk about TurboFlow technology. This involves patented extraction columns that rapidly remove a great deal of unwanted sample components, such as proteins, salts, and sugars. During the sample loading step, proteins are instantaneously excluded from the pores of the packing material. Of the molecules that enter the pores, only those that have an affinity to the surface chemistry are retained, while others are quickly eluted to waste. When we change the mobile phase to elute the analytes of interest to the HPLC column, we do this transfer in a way that we wind up with nice, sharp chromatographic peaks that the HPLC column produces. Any sample components that remain in the TurboFlow column after this transfer step gets washed away, while the analytes of interest undergo a final separation in the HPLC column and get eluted to the mass spectrometer. Whenever we add sample cleanup to an LC separation, the total runtime increases, but not the data window. The mass spectrometer sits there waiting for the next data window. That's the time it takes to measure all of the chromatographic peaks coming from our column. If the data window is half of the total runtime, then staggering injections into two channels will double the sample throughput and maximize the use of the mass spectrometer. This multi-channeling strategy works with any methods that elute the analytes of interest within a fraction of the total runtime, including LX methods that don't utilize TurboFlow sample cleanup. Cross-sequential operation maximizes the use of mass spectrometer's time, which a conventional HPLC system cannot do. Here is an example of analyzing urine specimens for benzodiazepines in one channel of the Prelude MD HPLC system, while the other channel analyzes the same sample for opiates. By multi-channeling across two channels, we are able to analyze two urine specimens for benzos and opiates in 13 minutes. That amounts to 18 injections per hour. Within an hour, we're capable of measuring 9 samples of benzos and 9 for opiates. The Prelude MD HPLC allows you to choose between TX mode, which utilizes TurboFlow sample cleanup prior to the final separation. The autosampler injects the sample into the loading pump's flow to the TurboFlow column. I'll show you the performance of a typical TX method used to measure appraisalism in synthetic serum that utilize protein precipitation for the sample preparation. In LX mode, and by the way, as you could tell from what I said previously, the T stands for TurboFlow. The L actually stands for laminar flow, which we consider conventional liquid chromatography, and the X actually stands for how many channels do you have. So in LX mode, the autosampler injects the sample directly into the eluting pump's flow into the HPLC or UHPLC column. And I'll show you the performance of a typical LX method used to measure reserpine and chloramphenicol in urine that has been diluted with mobile phase and then centrifuged. This data shows how reproducible the retention times were from the TurboFlow LCMS method for alprazolam during 2,000 injections over the course of 1,000 hours. The retention times across two channels were very reproducible, with RSDs typically around 1%. The retention times are typically within one standard deviation of the mean. Retention times for reserpine and chloramphenicol peaks from the LX method were also very precise. For these compounds, we get very little deviation from the mean. For applications such as these that don't require TurboFlow sample cleanup, we developed the Prelude LX4 MD HPLC. The Prelude LX4 MD HPLC is intended to separate drugs or compounds from a sample solution and introduce these separated drugs or compounds into a detector, and it is for in vitro diagnostic use. The Prelude LX4 MD HPLC has 4 parallel channels which can run identical or different methods simultaneously. Cross-Sequential Optimization maximizes the utilization of the mass spectrometer. If all channels run methods that have a total runtime of 4 minutes and data windows less than 1 minute, then a throughput of almost 60 injections per hour is achieved. In other words, the Prelude LX4 MD HPLC staggers injections across all 4 channels almost every minute under such methods. Here we see what the Endura MD mass spectrometer sees as it keeps up with each channel of the Prelude LX4 MD HPLC, running different methods across all 4 of its channels. In this case, the entire system achieves a throughput of about 22 injections per hour. Each channel has its own unique HPLC column and mobile phase, as well as unique data acquisition parameters for the mass spectrometer. However, each method must have the same ion source temperature so that there is no delay when switching between channels. The economic advantage of multi-channeled system is obvious. Rather than making a huge capital investment of 4 individual LCMS systems, which takes up a lot of space and resources, you can make a smaller investment for a single Prelude LX4 MD HPLC with Endura MD mass spectrometer. These systems, or one of these systems, fits in the space that would be taken up by a single LCMS system and uses the electrical connections, nitrogen gas, and argon gas of a single LCMS system. Yet, the Prelude LX4 Endura system can handle 4 times the workload of a single LCMS system. An additional solvent waste savings is realized by the unique design of Prelude HPLC systems. They utilize positive displacement pumps which stop to refill at the end of each analysis. Conventional reciprocating pumps needlessly run continuously and waste more solvent. Each channel produces reproducible results for individual analytes as we run 4 different methods simultaneously on the Prelude LX4 MD HPLC with Endura MD mass spectrometer. When measuring the concentrations of these analytes among 20 replicate injections, we typically get precisions less than 5% RSD with these performance tests. Running the same method across all 4 channels over a 24-hour period typically produces data showing that all 4 channels are virtually identical. These data also show how the Endura MD mass spectrometer kept up with the workload and produced precise area counts for each analyte. The Endura MD instrument is intended to identify and quantify inorganic and organic compounds in human specimens. It is for in vitro diagnostic use. The Endura MD is a triple quadrupole mass spectrometer designed to reliably identify and quantify various analytes from various samples that have been processed by various chromatographic and sample cleanup strategies in order to minimize sample matrix interferences. When coupled to optimized sample cleanup and chromatographic processes, the Endura MD provides accurate and reliable quantitation over a wide range of sample sizes and concentrations. The ClinQuan MD data processing software is intended for storing, retrieving, and processing laboratory data from the Endura MD mass spectrometer and Prelude MD HPLC instruments. The ClinQuan MD software is intended for in vitro diagnostic use. So the LC instruments and mass spectrometer work together through a single software which also processes the data and produces results. ClinQuan software streamlines the workflows for LC-MS applications in clinical laboratories. It features security functions which allow lab directors to set permission levels as required by the Clinical Laboratory Improvement Amendment, that's CLIA, of 1988. The software maintains audit trails to simplify record keeping and ensure the integrity of results. In summary, new multi-channel HPLC systems have been uniquely designed to address your concerns for efficient sample analysis, speed, throughput, and space restrictions. These Class 1 medical devices will help achieve the greatest efficiency for laboratory medicine. On behalf of my colleagues at Thermo Scientific, thank you for participating in this webinar. And I will now turn this back over to Trevor.
Thank you very much, Joe, and thank you all for sending in your questions. I encourage you to continue to do so throughout the presentations. If you joined us late, you can ask questions of our speakers simply by typing your query into the question box, which is located at the bottom of your screen. Our next speaker today joins us from EMD Millipore. Sean Crane is the Business Development Manager for EMD Millipore, focusing his attention to the in vitro clinical diagnostic marketplace. Sean has an education in biology and chemistry over 10 years, professional experience in the clinical diagnostic marketplace, working closely with pathologists, clinical biochemists, MLTs. He has a strong understanding of the medical laboratory needs, demands, workflow, validation, and accreditations. Sean has been working with EMD Millipore for 8 years and is well acquainted with the products and technologies of their portfolio. In addition to his work with EMD Millipore, Sean started his career as a medical laboratory technician and from there served to help launch a revolutionary HPLC-based qualitative urine albumin assay. Sean, thank you for joining us.
Thank you. Today I'd like to discuss the effects of bacterial contamination on an automated clinical chemistry analyzer and the prevention of such contamination. In the clinical laboratory, as we all know, water is a key factor. There are two significant reasons for the importance of water in the clinical setting. One is the need to comply with the norms or guidelines, such as those of the Clinical and Laboratory Standards Institute, or CLSI. And the second is the sensitivity of the chemistries themselves to water quality. This is a direct quote from a lab manager at the University of Pennsylvania School of Medicine who experienced contamination in her lab's analyzer. She did not take the necessary precautions when choosing a water purification system. So I will discuss the precautions that are needed later in this presentation. This slide shows a diagram of a water system feeding a clinical analyzer and 5 sampling points, S1 through S5, where the water has been tested for bacteria. The first sampling point is directly after the water purification unit, and the second is the internal reservoir of the analyzer itself. The remainder are located at sample delivery, reaction cuvettes, distribution needles. The chart in the lower left compares 2 hospitals, one with an issue of contamination and another without. The one with the issues had a microcount of 400 CFU per ml when the water was being delivered to the analyzer. Now, less than 10 CFU per ml is the CLSI standard for analyzer feed water, so they were way over that spec at 400. And as you can see, as a result of this water contamination, the analyzer quickly supported the growth of bacteria. By the time it reached the sample delivery, the count was too high to even test. The second hospital, by comparison, had 4 CFU per ml and was within the CLSI guideline of less than 10 CFU per ml, and as a result experienced stable counts in the analyzer. So this is just a good representation of how water quality can quickly affect the counts of bacteria in an analyzer. And as we all know, if there's bacteria in an analyzer, there's going to be problems. Decontamination of the analyzer itself will have to ensue. So what types of bacteria are found in pure water? What technologies are designed for the prevention? Typical strains of gram-negative bacteria adapted to stringent conditions such as the virtually nutrient-free environment of pure water are listed here. These nasty strains have been identified by genome sequencing, and prominent offenders found most often as a source of contamination in clinical analyzers. So we're dealing with a particularly robust bunch here. So the effects of bacterial contamination on clinical water include direct interference with some assays, as well as some indirect effects. So directly speaking, we can see turbidimetric measurement impacts of the microorganisms themselves. So in effect, they're acting as particles. Indirectly, the proteins, enzymes, small organic acids released by the bacteria interfere with the several assays. So it's not just the bacteria themselves that can affect the assays, but their byproducts themselves. The byproducts of the bacteria magnify the hindrance of the bacteria. So typical impacts of bacterial contamination include unstable calibrations, high absorbance of blanks, reference drifts, errors on mean patient values. So if one or more of these aberrations are observed, one should look to bacterial contamination as a source of the problem. In the first place, one should investigate is the water source. If we take the calcium assay as an example, it's a very popular assay, as we all know. Issues with calibration standards and or mean patient values can be observed, and typically decontamination solves the issue. So I'm going to look at the calcium assay in more detail. So as many of you are aware, the calcium procedure is based on calcium ions reacting with the arsenazo complex to form an intense purple colored complex. So this is the basic concept. And one would assume that calcium ions present in the pure water would be the source of the problems, but water with a resistivity of only 1 megaohm, or in other words, it's got practically the CLSI guidelines call for less than 5 megohms or greater than less than 15 megohms. So even water that's not even within spec of the CLSI guidelines would have a maximum level of calcium at 500 ppb, but the contribution of the calcium from the water, even at that resistivity, is below 1% of the concentration of the calcium measured. So even when we have that type of calcium present in the water, it's really just a small percentage of the total that's being measured by the assay. Therefore, while the analyte being measured is an ion, the ionic purity of the water is not the source of the problems observed with the assay. So this is a popular misconception. And second. So other contaminants potentially present in the water can bring some light to the subject and enable us to rationalize where the problems are coming from. So if the organic level in the water has not been reduced correctly, small organic acids, including and similar to oxalic acid, which can be released from bacteria, can be present and bind the calcium, thus reducing the apparent concentration of calcium. Small organics are byproducts of bacteria. So similarly, amines, similar in structure to the arseniazo complex, may also bind to the calcium ion. Amines are also byproducts of bacterial contamination. Finally, high bacteria level in the water will generate the presence of proteins, significant levels of proteins that will complex with the calcium, therefore leading to an apparently low level of calcium as well as erroneous values. So as I mentioned, bacteria releases organic acids, amines, and proteins, all which can interfere with the assay itself. It's not the calcium. So it's important to consider the overall purity of the water and not to focus on just one quality parameter only. It's a popular misconception in a clinical laboratory that if their resistivity on their water system is within spec, that the entire water system is in spec. But one needs to really look at the overall picture and look at other contaminants such as bacteria and possibly organics and other byproducts of bacteria in the water. All right, so let's look at how to prevent bacterial growth in water used in the clinical analyzers. Technologies include reverse osmosis, UV germicidal lamps at 254 nanometers, and the 0.22 micron screen filters embedded in the complete water purification chain. So the purification of water is a three-phase process. If we want to look at water purification system, it's really three phases. Broken down, the first phase is the slowest phase, and that's where the bulk of the contaminants of water are removed. That water is typically that's used reverse osmosis, electronic deionization, and then that water is stored in a reservoir, which can be considered the second phase. As the water is needed, the third phase is the delivery of the water from the reservoir to the analyzer. And in that delivery, it gets polished further, it recirculates, and it passes through a 0.22 final filter. So we typically think of it as a three-phase process. And this slide shows the purification chain in more detail. And this chart on the bottom lists the technologies and the contaminants in the water that each removes. As you can see, there's no silver bullet, and the only technologies effective at removing bacteria are reverse osmosis, UV, and 0.22 micron filtration, screen filter. So it's important to point out that one of these alone will not bring the bacteria levels down to less than 10 CFU per ml, but the combination of them must be used to keep to that standard. A properly designed water purification system will incorporate reverse osmosis as a means to remove 95% of all bacteria from the tap water feed at the first step. Equally important is a 254 nanometer UV in all three phases. So we have one in the purification step, in the storage reservoir itself, and one in the delivery or distribution. Other technologies commercially offered are not suited to control bacteria over time. And these include DI resin tanks, which are actually allow water to remain stagnant in them over time, promoting a perfect environment for microbes to proliferate. Furthermore, the design of such systems utilize just a single UV lamp. So what are the risks and outcomes of bacterial contamination of an analyzer as a result of improperly designed water purification systems? Some of you, or know anyone that's ever had an issue with an analyzer and bacteria, have noticed one or all of these. So it's erroneous patient value results, cost associated with repeat running of calibrations, so time spent running repeat calibrations, and the cost of the reagents to run them, and the potential impact on laboratory accreditation and reputation, and decontamination of an analyzer, which is considerable downtime. It could be an entire day to decontaminate an analyzer, and then all the costs associated with that. So EMD Millipore does not cut corners when designing our water purification units, and we take bacteria removal and mitigation in pure water with critical importance, especially in our systems designed for the biomedical laboratory. Our systems are unmatched in this regard. And just another reason why we're considered the top tier. So this slide just lists some of the considerations we take into account when designing a system for an analyzer. So what are the takeaways? Water impacts clinical assays in many ways. In the clinical laboratory, a good practice is to consider water as a critical reagent. Therefore, care should be given to the quality of the water required to run various assays in the laboratory and to water handling in general. Designing, selecting, and maintaining water purification units in a suitable manner can reduce issues linked to water quality and clinical assays, instrument downtime. Thank you for your attention. If you have any questions, please feel free to chime in.
Great, thank you very much, Sean. And I believe we will move into the question and answer session now. We do have a number of questions that have come in from the audience. And Joe, if you've joined us again, perhaps I'll direct the first question at you.
Sure.
Regarding the TurboFlow technology, how well do you think that would work with decomposed post-mortem samples?
Well, that's an interesting thing that you mentioned because we have been speaking to some customers analyzing such samples for various drugs. And from what I understand from these customers, it does a good job of removing any of the sample matrix interferences that would cause a problem in estimating the amount of these compounds, because the TurboFlow technology is designed to remove unwanted components such as proteins, salts, and sugars, and very often we can also remove very hydrophobic compounds such as phospholipids, depending on the compounds that you're trying to analyze. So it is very useful for that kind of an application.
Thank you very much. Sean, the next question I'll direct to you. The bacteria that you mentioned, what are they typically feeding on?
If you look at organics in the water, so the CLSI guidelines is less than 50 ppb for organics in the water. They'll be feeding on trace organics, some trace minerals as well.
Great, thank you. Now, Joe, could you explain the difference between HPLC and UHPLC?
Yes, typically HPLC is done using columns packed with particles 3 micron in size and larger, and they typically are operated at pressures around 200 bar or less, whereas UHPLC operates at much higher pressures because those columns are typically packed with particles smaller than 3 micron, typically less than 2 micron. So that means that if you're going to do UHPLC, you would need to have pumps as well as all of the connections, valves, and so on, capable of handling such high pressures. Our systems typically can go up to 1,000 bar.
Great, thank you. Now, Sean, what are the methods for testing water for bacteria?
Well, all of EMD Millipore's systems are equipped with a sanitary sampling port. So why that's important is because you don't want to have a valve that is contaminated with bacteria, that you're taking water out. You're removing water from the system with such a dirty valve, because then you'd just be getting counts that aren't really indicative of the system itself. So our systems include what's called an ESP sanitary sampling port. So it's a zero dead leg valve that actually allows you to sample water from in line of the system. So you're getting a true representation of the water, and then that water can be sent to a micro department for a micro count.
Thank you. Now, Joe, could you explain what the TX and LX stand for?
Yeah, I actually mentioned this very briefly in my talk, but these are marketing terms, and the T refers to the plumbing in our systems that does include the TurboFlow online sample cleanup, and that plumbing allows the autosampler to squirt the sample to the TurboFlow column, where we do online extraction and elution to an HPLC or UHPLC column. Whereas the L, that stands for laminar flow, which we consider to be conventional liquid chromatography. And that plumbing allows the autosampler to squirt the sample directly to the HPLC or UHPLC column for final separation and elution to the mass spectrometer. The X stands for how many channels do you have, because not only are we well known for doing the online sample throughput or online sample cleanup through TurboFlow technology, we're also well known for improving sample throughput because of our multi-channel systems.
Great, thank you. And while I have you, how often does the TurboFlow and HPLC columns need to be replaced when running typical batches?
Most of our customers are able to run these TurboFlow columns for at least 1,000 injections, and many are able to get away with running 2,000 or 3,000 injections, depending on what sample prep is done prior to the injection. And the fact that the methods are designed to backflush the TurboFlow columns and clean them out thoroughly with mixtures of solvents, we are able to regenerate these columns between injections and ensure that we avoid carryover and get them to last as much as possible. But very generally, around 1 in 2,000 injections is very typical for most methods.
Great, thank you. Now, Sean, do the water purification units require any sort of sanitization?
All of our systems are sanitized at start during installation, so that comes standard with the system as we do a startup sanitization. After that, it's not necessarily required, but it's recommended that the systems be sanitized on an annual basis, just to prevent, you might have a very small colony of bacteria somewhere within the system that you might not necessarily be picking up in any of your micro counts. So if it goes unnoticed, it could present a problem down the road. You might be running fine for a number of years, and then all at once you might see a huge spike, logarithmic growth of the bacteria, and that could then in turn contaminate your analyzer, and that's when you start to see all those problems. So it could kind of come up without warning. So we do recommend an annual sanitization of the water system, and we provide that with our service plans.
Excellent, thank you. Now, Joe, is it necessary to run all calibrators and QCs on each channel of a multi-channel system?
Typically, our customers do need to demonstrate that each channel of a multi-channel system is identical to the others. And they'll have plenty of data to show that they can run all calibrators and QCs on one channel and justify not having to do that on a routine basis. So all of that sort of experiment is done during the validation process. And once they have enough data accumulated to demonstrate that all channels are equivalent, they typically can just go ahead and run calibrators across the channels. However, it is recommended and for some companies required to make sure that each channel does get to run all the QCs at least once or twice in large batches in order to validate the data from each channel.
Excellent, thank you. And Sean, in your experience, are there any water purification technologies or designs that are particularly prone to promoting bacteria in the water that's produced?
Yes. Well, there's what's considered what's called service DI, which are basically tanks with regenerated deionization resins. What that technology incorporates is it's one just dead leg, meaning it's a one-way system to feed an analyzer, and there are these tanks that remove ions out of the water. I mentioned this briefly in the presentation. So with those tanks, when the water is not being called for by the analyzer, water is just sitting stagnant in them. And it's a perfect place for bacteria to grow. In addition, the resins on the beads inside those tanks that are regenerated with acids, heavy acids and bases. So some of those actually leach out of the water off the beads, and it gives more nutrients for the bacteria to grow on. So those tanks do a really great job at removing ions, but in terms of removing bacteria and organics, not a very good idea to use those in a clinical lab.
Great, thank you. And Joe, could you explain how positive displacement pumps work?
Yeah, the positive displacement pumps, unlike reciprocating pumps, which are essentially pistons that keep going ka-chunk, ka-chunk, ka-chunk all the time, and using check valves to ensure that flow goes only in one direction, even at high pressures. The positive displacement pumps do resemble a syringe, which fill at the beginning of the analysis and slowly squirt out at a very controlled flow rate during the chromatography. So we have to make sure that our methods are designed to not run out of the solvent in these pistons or syringes in the positive displacement design. And each one of these pumps holds about 300 milliliters of solvent. And that's typically plenty to achieve the separations that we achieve with our methods. So at the end of the analysis, the flow stops and the pumps essentially refill and get ready for the next injection, rather than keep pumping continuously like reciprocating pumps do. Therefore, with this type of design, we avoid wasting so much solvent between injections.
Great, thank you very much. And as I look at our question board, it looks like we've gotten through just about all of our questions. I do have some requests for some further information that I'm going to forward to you. And with that, that brings us to the end of today's webinar. If you have any further questions, please reach out to our presenters directly. Their contact information, as you can see, is available on the screen now. And just a reminder that today's webinar video will be available at the link you see on screen now. On behalf of Lab Manager, I'd like to thank our panelists today for the hard work they put into their presentations. And I'd also like to thank our sponsors, Thermo Scientific and EMD Millipore, for supporting our Tech Trends webinar series. As well, I'd like to thank all of you for taking time out of your busy schedules to join us today. Please mark your calendar for our next Tech Trends webinar, which will focus on forensic science techniques. This webinar will be taking place Thursday, October 15th from noon until 1:30 p.m. Eastern Standard Time. For more information on this and other webinars, please visit our website at www.labmanager.com. We hope you can join us again next time. Thank you and have a great day.